Inversion, and let's understand in insertion and deletion once again. So what is insertion and deletion? In the last class I told you frameshift mutation. So this topic is the frameshift mutation. This is inversion, this is substitution, and this is frameshift mutation. Now frameshift mutation is what? Imagine there's a long sequence of DNA. If you insert something here or insert anywhere, we all know codons have to be read in continuous manner, 3, 3, 3 altogether. If you add something here, from the point of entry, reading is going to change, because now you are going to add one more nucleotide. So once you add another nucleotide, example ACCTATTG, this is the original reading. If you insert an A here, then the reading now will be AAC. So this has gone to the next codon. Then the next codon will be CTA, and next codon will be TTT. That means complete change in the information. So from the point of insertion, the frame of the reading has changed or shifted. That's why it is called as frame shift mutation, and this will cause complete change in the amino acids from the point of insertion. Before the point of insertion, remember DNA is a long piece. So before the point of insertion, DNA information remains same, but after the point of insertion, information will change. Same manner, deletion also. If you remove one base from here, then the reading is going to change. Like if you remove C from here, ACT, now the information has again changed. So information change will cause change in the protein formation, because different amino acids will be coded. Remember, it is even possible a mutation can cause a stop codon to come. A stop codon will not cause protein formation, because its information is to stop the process of formation of protein. How many stop codons are there? UAA, UAG, UGA. In the last class from that NCERT checker box, I told you this point. Now let's understand what may happen if there is addition of more or deletion of more. Addition of one base and deletion of one base. Now imagine two things are occurring. One is moving out and the other is moving in. Will they bring any change? There will not be any frame shift mutation, but there will be a mutation. Imagine that if you insert a T here, and if you remove the A from here, then obviously you are adding one and removing one, so there will not be any frame shift, because because one you have added, one you have removed. But the problem is that information has changed. Because information has changed, so it will cause a mutation, but it will not be a frame shift mutation. Another point, it is possible insertion of two or deletion of two base. Now when you add two bases together, or when you remove two bases together, that will also cause frame shift mutation. Another point, if you insert three bases together, that means you are adding one new codon. Or if you are removing three bases together, that means you are removing one codon, then there will not be any frame shift mutation, but yes, there will be a normal mutation. Normal mutation means information of the DNA has changed. Now accordingly, there are three types of this. I have already covered before if you have gone through the notes carefully. If there are three types of mutations possible according to this information: nonsense, missense, and same sense mutation. Let's understand quickly. It's an easy concept. What is nonsense mutation? In DNA, if the information is ATT, ATC, ACT, then what will happen? If these are the informations formed because of mutation, then ATT will always cause formation on the mRNA UAA, and on the mRNA UAG, on the mRNA UGA. And from that checker box of yesterday's class, we learned that UAA, UAG, UGA are stop codon. So whenever a mutation stops the protein formation, it is called as non-sense mutation. This mutation does not bring any sense, and protein formation has stopped. Now in the same manner, there is a possibility a missense mutation. What is missense mutation? The DNA's information is GAT. Now imagine for a second it is changed to GGT. Now if it was GAT, the mRNA should have carried the information of CUA, and this codes for leucine, one amino acid. Now if GAT is mutating into GGT, then GGT will code for which codon of mRNA or information of mRNA? CCA. Now CCA codes for proline. All these things are in that checker box of yesterday. So this will cause the carrying of information for proline. So leucine amino acid is replaced by proline. Now this means another information. So what is missense mutation? When the DNA mutation which does not cause the formation of the stop codon but will cause another mRNA to be formed which will code for another amino acid. So that means there is a change in the expression, and protein quality may be changed, may not be changed. Now what is same sense mutation? Sometimes DNA's mutation does not bring any change in the amino acid. For example, if the DNA carries the information TTT, then the mRNA's information should be AAA. AAA will code for lysine. Now if this TTT is replaced by, so a mutation at the third position, TTT is replaced by TTC, though now TTC will cause formation of AAG type of mRNA, but if you check the checker box, this also codes for lysine. That means there are sometimes mutation which will not bring any change, and such mutations are called as same sense mutation. I hope this concept is clear, that the mutations can be non-sense mutation, missense mutation and same sense mutation.